Okay. Yep. Hello, I am a PhD student at Wayne State University in Detroit, and thank you all for being here. I know you have a choice this year, and thanks to the organizers for the opportunity to present my work. So if we consider disease traits, we know that there's a mismatch that exists between the genotype and the environment that may be responsible for a higher disease risk. Studying gene-environment interactions at the organismal level, though, can be fairly complicated, and it presents a lot of challenges, such as environmental covariates that tend to be very complex and can be hard to measure and control for. So alternatively, we can focus more on specific cellular environments and kind of use them as a proxy for the whole organismal environment. And in doing so, you can use a wide panel of, say, treatments in vitro on specific cell types and then measure the resulting molecular phenotypes. And so even though these represent simplified versions of the organismal environment, the advantage is that you get tight control of the environment by treating the cells at specific times and specific concentrations. And then you can take those interactions that you observe from the cellular environments and kind of use those to get a better understanding of the organismal, or of variation amongst complex traits. One method that's been used so far to identify gene-environment interactions is through in vitro studies doing response eQTL mapping. There's a few studies that have done this so far, and they do it in a one treatment or maybe a handful of treatments. But the problem is that it doesn't scale very well if you want to look at a very large panel of treatments of different environments. For example, if you want to do 50 environments and 100 individuals, you do 5,000 experiments, which is kind of intractable. So we need a different approach. So allele-specific approaches then are an alternative to eQTL mapping that can identify eQTLs within individual samples at heterozygous sites. Through the allele-specific expression, it's possible to identify genes that have kind of eQTLs when observing a departure from the 1-to-1 ratio in the transcript, the reads covering a variant in the transcript. One key advantage of using allele-specific analysis, of course, is that you're controlling kind of some of the confounders because you're using the same individual. All the sample, or all the data that you're looking for comes from one individual. So then to identify genes with gene-environment interaction QTLs, you can then contrast the ASC pattern across two different environments, shown here as environment X and environment Y. And so essentially, a gene with a GWE-eQTL is one that shows a response in X, but not in Y. And you only see the ASC in X. So, as the talk title says, we did this in 250 cellular environments, and here's kind of an overview of the compounds that we use. There's 50 of them that kind of filter into these six broad categories. There are things like dexamethasone. We use a lot of common drugs like aspirin. We also have some environmental contaminants like heavy metals, or not heavy metals, sorry, metal ions. And we actually use polluted air from Detroit called particulate matter 2.5. And then we have 5 different cell types that we use, all of which are primary except for the LCL lines. And they all have, at the very least, related cell types in ENCODE if we wanted to dig into some of the other data. So overall, kind of the pairwise, this gives us our 250 cellular environments. And what we can do is measure the gene expression response in each of these cell types in response to each of these compounds. And so here's a heat map that shows the number of differentially expressed genes in each of these combinations, where the darker the red, the more genes are differentially expressed between a treatment and its matched control. And so you can see that there's kind of a wide range of treatments that do respond and treatments that don't. Some have a response like vitamin A across all of the cells that we looked at. Others are more cell type specific. So then what we do is we take the strongest responses and we follow up on them. We do a deeper sequencing to really get at those kind of in detail, the differences between the transcriptional responses. And here's an example of a PCA for one cell type. This is PBMCs, and we have 3 different individuals shown with the different shapes, and then each of the treatments that we used on this cell type are shown in the different colors. And you can see first that the individuals cluster very nicely, and then you also see nice groupings of kind of biologically similar treatments like zinc and copper, these 2 metal ions. And we see similar results if you do hierarchical clustering and so on. Then for heterozygous variants, we can examine rho, which is the proportion of the reference allele over the total reads, and then calculate the log odds of ASC, shown here as the beta. And to do this, we use a method that our lab developed called Quasar. So what we see is across 48 environmental contexts that we deep sequenced, we see 6,000 examples of significant allele-specific expression, and this corresponds to about 4,000 unique ASC genes. And then digging in a little bit more, for any given individual, what we see is on average about 0.5% of any gene that has a het is an ASC gene, 0.5%. So then to identify genes with gene environment ASC, we used MESH, which was developed by one of our collaborators at the University of Michigan, which uses a Bayesian approach to contrast 4 possible configurations. Either there's no ASC in either the treatment or the control, there's ASC in both of the conditions, there's ASC in treatment only, or there's ASC in the control only. And so this plot shows the evidence for ASC. Shown here is the control or the treatment z-score, and it's colored based on those MESH categorizations. So black here shows the evidence for shared ASC, and what we see is consistent with what's been reported in the literature so far, which is that most ASC is shared across treatment and control. But we still are able to identify examples of treatment-only ASC shown in the red, and the control-only shown in the blue. And overall, we see 141 control-only ASC examples and 141 treatment-only. So here's some of the more striking examples of the treatment-only ASC. And shown here is, again, the beta for ASC. And in red shows the treatment betas, and in black shows the control, where the error bars represent the 95% confidence interval. And even though ASC here is determined within individual samples separately, we actually see really good concordance for some of the conditional ASC across different treatments. So for example, here's kind of the same signal shown in LCLs for perfluoric acid and for selenium. And another example between iron and caffeine and PBMCs. So then, given the large number of environmental conditions that we're investigating, we can now ask which factors contribute to the different ASC configurations. And to do that, we used a multinomial model to predict ASC in each of the categories from MESH, where the default would be no ASC, and then we can look for shared control-only or treatment-only. And to do this, we used a model with elastic net variable selection, which is implemented in the GLMnet package in R. And so overall, the factors that we consider are the cell type, the treatment type, the gene expressions, like the epikms, and the differential expression log-fold change. And this plot shows, so here's the factors that we were considering, and then here's essentially how the different factors influence the odds of the 3 configurations shown. And so what we find is that some treatments do increase the likelihood of having condition-specific ASC. For example, caffeine here has higher relative odds for treatment only, whereas retinoic acid shows higher relative odds for control only. And overall, the most relevant factor that comes out is the differential expression, where what we see is, or for treatment only ASC, and what we see is that the odds of the treatment only ASC are 2.2-fold higher for every 2-fold increase in gene expression. And we can actually see this pattern looking at it a slightly different way. So when we compare ASC in the treatment to the one in the matched control using kind of a frequentist approach, where here this is a QQ plot where the p-values are p-values for the difference in ASC between treatment and control. And if you look at genes, so here they're colored by expression level in the treatment and in the control. And the green and the blue show high in both and low in both, respectively. The gold here shows high in the control but low in the treatment. And those are all about the same, kind of have the same evidence for ASC. But what you see is when you have overexpression in the treatment relative to the control, you have an enrichment for this ASC signal. And so finally, we can use some of these examples of GWAS interactions identified in this in vitro system to learn more about the organismal level GWAS interactions. And to do this, we first started looking at the overlap between GWAS variants and these variants that we've identified. And so this is a little preliminary, but essentially the hypothesis is that just by doing this kind of overlap, we can provide some putative molecular mechanisms behind the observed risk or protective environmental factors in these complex traits. So one example here is this long non-coding RNA that has allele-specific expression in response to retinoic acid, and it's linked to obesity-related traits. And some molecular studies have been showing that a reduction in diet-induced obesity in mice treated with retinoic acid. Another example here is this treatment-only allele-specific expression shown in response to caffeine in smooth muscle cells in the gene TIMMDC1. This gene contains variants that are associated with MS and with primary biliary cirrhosis, and recent studies have suggested that caffeine actually has a protective effect on both of those diseases. And so currently we're working on kind of a more comprehensive way to put together all these individual examples. So in conclusion, I didn't really talk much about the approach, but we have this new high-throughput approach for GWAS identification across hundreds of conditions, hundreds of environments. What we see is that most ASC is shared across the conditions, but we can identify quite a bit of those GWAS interactions. Most significantly, I think, is that genes that are differentially expressed are 2.2 times more likely to show extreme GWAS ASC, which is really neat, and that these GWAS interactions detected in vitro can hopefully help inform complex trait variation. And I'd like to thank my two PIs, Francesca and Roger, and everybody in our lab, our collaborators, and my school, and our funding sources. I'm happy to take any questions.
Go back to your first heat map of the genes that show response to the treatment
Sure.
in the 5 different cell lines.
Where is it? Oop, too far.
So
There we go. Okay.
So it really seems there's kind of a large response, a zero response, and then kind of an intermediate response, right? And can you make sense of kind of which combinations of cell lines and responses fall into that class?
Yeah. So a lot of the zero response comes from, well, essentially just things that aren't known to respond. I mean, the way we set this up at the shallow level is just to, or things that aren't, I shouldn't say aren't known to respond, but maybe there wasn't specific evidence for it, but it was included on our panel because, you know, in one of the cell types, maybe it was, and maybe another it wasn't. But really, it's a shallow pass to see what's responding and what's not. And then we choose to follow up and spend our resources on the things that do have a response. Part of that comes from the fact that there's, for a lot of these, conflicting literature about what does and what does not respond. And this is a quick first step. So then we can safely ignore things that we see, okay, they don't respond, so let's not worry about following up on them. On the other end of the spectrum, we do know that some of the things that have an extremely large response, for example, iron, might do so because it has kind of a cytotoxic effect. And so there are cases where we don't follow up on iron, or we exclude it because it ends up killing the cells, and that's why you see such an exaggerated response. Not for all of them per se, but that's one example.
And all this is going to be dose dependent, right? So
Yeah. So we would expect it to be dose dependent, but for all of these compounds, we use the Mayo Clinic kind of recommendation, or the reference ranges for what's noticed in blood. And I should mention that all of these are chosen to be bioactive in blood, not needing to be metabolized in the liver or anything. So we do choose what concentration should be kind of physiologically relevant when we treat the cells.